Hi folks, and welcome to another edition of Zog Science. Today we're going to be talking about DNA replication and chromosome structure. So, DNA replication and repair is the result of many proteins that are working together, and the relationship between the structure and function of DNA is shown in its double helix. And Watson and Crick were, when they discovered the structure of DNA, suggested that the base pairing was what would allow for the copying mechanism of the genetic material, or of DNA. And so the way that this works is that you have the original parent molecule, the molecules then separate out, and you add on the accompanying complementary strands of DNA. So we can use the G in order to know that that's going to pair with a C. And so now if we compare our daughter molecules, the ones that are being produced, they are equivalent to the parent molecule. Because the two strands are complementary, each one is going to act as a template for building the new strands of DNA in replication. And in DNA replication, the parent molecules unwind, and then they are going to be built. And we're going to look at what all of the proteins that are involved in how that process works in just a little bit. This model is called the semi-conservative model, in that the new molecules are made up of half of the old strand and half of a new strand. There were other models for how whether or not this is the correct model when this was first proposed. So there's the conservative model, which is that the molecule of DNA, the new molecules are going to be all old DNA and then all new DNA. Then you have the semi-conservative one. And then you have the dispersive model, which is that you just kind of have random amounts of DNA that end up in each of the new molecules. And there were experiments that were performed by Messelson and Stahl that eventually supported the semi-conservative model. So what they did was that they grew bacteria with a heavy isotope to begin with of nitrogen. So the DNA was heavier. They then transferred those bacteria into a medium with a lighter isotope so that all of the daughter cells would have the lighter isotope. And so what they did was that after the first replication, they saw that all of the DNA was in one place, which supports the semi-conservative model. It does not support the conservative model because you would expect to see both available, because you should see new DNA and old DNA. Then after the second replication, they saw that they began to see some of the DNA being made with only the lighter isotope, which if we go back to our original semi-conservative approach, we would see, right? We would see some of the DNA being made with only new stuff, but that does not work with the dispersive approach, right? And so the semi-conservative model was confirmed through these experiments. Now, the copying of DNA is pretty fast. It happens at a very fast rate, and it's also remarkable in terms of the accuracy. There are dozens of enzymes and proteins that participate in DNA replication. Sometimes it's called a replication machine, and it is much more is known about that in bacteria as compared to eukaryotes, but most of the process is similar between the two. Replication begins at sites called origins of replication. Essentially, that's kind of places where the two DNA strands are separated, which is opening up a replication bubble. And at each end of the bubble is a replication fork, which is a Y-shaped region where the parental strands of the DNA are being unwound and then the daughter strands are being created.
Replication of a DNA molecule begins with a specific sequence called an origin of replication. The two strands separate, forming a small replication bubble. DNA replication starts and then proceeds in both directions until the original DNA molecule is completely replicated. Each of the two daughter molecules consists of one old strand, shown in purple, and one new strand, shown in green. The DNA of a eukaryotic chromosome has multiple origins of replication. The multiple replication bubbles allowed this very long molecule to be copied quite quickly, resulting in two daughter DNA molecules.
So this is a model showing you what is happening at the replication forks. So you have topoisomerase, which is helping to stabilize the DNA molecule because it is twisted. And so topoisomerase is there to help to untwist it. Then you have helicase, which is breaking the hydrogen bonds that are between the nucleotides of the two strands. Then you have some single-strand binding proteins. These proteins are stabilizing the fact that DNA wants to be double-stranded, doesn't want to be single-stranded. So these single-strand binding proteins are helping to maintain that stability. And then you have primase, which is adding an RNA primer. And that RNA primer is incredibly important because without it, you cannot actually begin replication. And so again, here's just kind of what those different proteins are, what their enzymes are, and what they're doing. Now, in E. coli cells, as was mentioned in that video, you have a replication fork with the bubbles, and those are going to be splitting apart. And so those bubbles are going to be going around the circle, and eventually they meet on the other side, and so you end up with your two new daughter molecules. Here's an actual picture showing that process happening. You can see how right here, these are our replication forks, and they are moving around the circular DNA. In eukaryotic cells, you get multiple bubbles that are occurring along the linear DNA. Remember that prokaryotic DNA is circular, eukaryotic DNA is linear. And so you get multiple of the replication forks and bubbles that are then going to meet up together, and you can see those here. Now, in order to synthesize a new DNA strand, we have to have DNA polymerase. The DNA polymerase is the enzyme that is able to add on new nucleotides. However, it cannot initiate synthesis of a nucleotide by itself. It can only add nucleotides to an existing chain. And so the initial nucleotide strand is created by that RNA primer that is going to put the little strand of RNA, and then the DNA polymerases can begin adding onto that. And the rate of elongation is about 500 nucleotides per second in bacteria and about 50 per second in human cells. So that's very, very fast. Each nucleotide that is added to the growing DNA chain consists of the sugar attached to a base and 3 phosphate groups. So this is very similar to ATP, except that you have different ones for each of the nucleotides. So you have like a DATP, you have a DTTP, you have a DCTP, and a DGTP. And so those are all used. The energy is kind of included within the molecule itself because it has those extra phosphate groups. As each of the monomers is added on, the phosphate groups are broken off, and that free energy that is released is used to catalyze the reaction, or not to catalyze the reaction, but is used to power the reaction. So here's our nucleotide with its 3 phosphates. The 2 phosphates are popped off, and the nucleotide is then going to be added on to the growing DNA strand. Now, newly replicated DNA strands are formed anti-parallel to the template strand. DNA polymerases add nucleotides only to the free 3' end of a growing strand. Okay, so therefore, new DNA strands only grow in the 5' to 3' direction. And the reason why is that this 3' end, there is a hydroxide group, and this is a dehydration synthesis reaction. So this hydroxide group is going to be combining with a hydrogen from the phosphate group. They're going to come off and form water, and then the phosphates are going to create the bond. The 3' end, remember, is due to the fact that this is the 3' carbon. The 5' end is due to this carbon right here being the 5' carbon on the deoxyribose sugar. Now, there are 2 different kind of strands that are occurring at both times at the replication fork. Remember, it's a Y-shaped structure, so you're getting 2 new strands. One is called the leading strand, and the other one is called the lagging strand. So let's take a look a little bit more closely at what's going on with the leading strand.
Let's now look at the process of DNA replication more closely, starting at an origin of replication. The DNA opens up there to form a small bubble. Molecules of an enzyme called helicase attach to the DNA at the ends of the bubble and continue the unwinding of the double helix. We'll focus on one end of the bubble, on the Y-shaped region called a replication fork. The 2 strands would naturally tend to rewind, that are held apart by molecules of a single-strand binding protein. The synthesis of a new strand begins when an enzyme called primase attaches and synthesizes a short RNA strand that is complementary to one of the DNA strands. This piece of RNA is a primer. DNA polymerase then adds DNA nucleotides to the 3' end of the primer. It continues to lengthen the new DNA strand by adding nucleotides complementary to the template strand. Notice that DNA synthesis always proceeds in a 5' to 3' direction. The strand just made here is called the leading strand. For clarity, we'll keep the new double-stranded DNA untwisted.
So that leading strand, notice that it is moving towards the replication fork, and therefore it is able to synthesize continuously. The lagging strand, which we'll take a look at in a little bit, is not able to synthesize continuously. So on our leading strand, we have our RNA primer that's added on by the RNA primase. The DNA polymerase is then going to begin adding on the nucleotides, moving in a 5' to 3' direction, which is then going to be able to replicate continuously as helicase is unzipping the original parent DNA. Now, the lagging strand, the DNA polymerases necessarily must replicate in the opposite direction because, remember, it can only add on to the 3' end. The other strand of DNA, right, it's going to be 5' here and 3' in going away from the origin of replication. And away from the where the helicase is unzipping. And because of that, you're going to get to the end of that, and then you need to add on more nucleotides back towards the origin of replication. And because of that, you get little segments of the lagging strand, which are called Okazaki fragments. And after formation, DNA polymerase 1 is going to come in and basically kind of remove the RNA primers and then replace the nucleotides. And then ligase is going to come in and actually fill in the gaps because, again, you're kind of replicating these discrete units. You got to come in and fix that process.
The other new strand is called the lagging strand. Unlike the leading strand, the lagging strand cannot be made continuously because DNA polymerase can only add nucleotides at the 3' end. Instead, the lagging strand must be made of fragments that are linked together. Despite their names, the leading and lagging strands are actually made at the same time. For clarity, here we will just show the lagging strand being pieced together. We'll start with a brief overview of fragments being formed and linked. First, an RNA primer, shown here in red, forms the beginning of each fragment. The rest of each fragment is then synthesized from DNA nucleotides in a 5' to 3' direction. The resulting segments are called Okazaki fragments. Next, each RNA primer is replaced with DNA. Then the gaps are closed to form the lagging strand.
So this diagram shows you the process that we just looked at, but slowed down a little bit. So we've got our RNA primer that is being added on by primase. We then have our DNA polymerase coming in and adding on the nucleotides until it reaches the end of this segment. RNA polymerase is going to pop off. You then have a new primer added on, and then the RNA polymerase or DNA polymerase is going to add on the nucleotides again to get to the next RNA primer. While that's happening, DNA polymerase 1 is replacing the RNA with DNA, and then DNA ligase is going to fill in the gaps. And so because this process is taking place over time, it's called, and you're not able to just replicate continuously, it's called the lag. Now, the process of DNA replication is generally very accurate. However, there are times where that process does not take place correctly. And so in mismatch repair of DNA, other enzymes are going to come in and correct things. A hereditary defect in one of those enzymes that does the correcting is associated with a form of colon cancer. And so because of that, this defect is going to allow basically cancer-causing errors to accumulate faster than they normally would. And DNA can also be exposed to harmful chemical or physical agents, right? So either stuff like UV damage or cigarette smoke, X-rays, things like that. These can also be repaired in nucleotide excision repair. A molecule called a nuclease cuts out the damage and then replaces it with the correct sequence. So, for example, what can happen is that actually you can have molecules that form in bonds to each other. The nucleotides actually attach to each other. So a nuclease will come in, it will cut that out, and then it will replace the nucleotides that should be there. Now, the error rate of proofreading repair is very low, right? But it's not zero. So sometimes changes in nucleotides become permanent, and these changes or mutations are the source of genetic variation upon which natural selection generally operates. Not all of natural selection operates based off of mutations, but much of it does. And the linear DNA, the replication machinery, cannot complete the 5' ends of the daughter strands because, again, you have that RNA primer at the end. And so that RNA primer can't be changed into DNA because DNA polymerase 1 doesn't have the ability to replace that because it doesn't have anything to build off of. So repeated rounds of replication produce shorter DNA molecules for your eukaryotic DNA. And so at the ends of your chromosomes, you have these special sequences called telomeres. And telomeres are located at the end of each chromosome. They're shown here in the red. And what they are is they are multiple repetitions of one short nucleotide sequence, and they don't have a purpose other than just to slow down the shortening of the DNA molecules to where the DNA molecules have actual genes. So essentially, they're kind of like just areas that can be cut off at the ends, so that way nothing bad happens. It has been proposed that the shortening of telomeres is connected to aging, and the chromosomes of germ cells have, or the cells of germ cells have a special enzyme called telomerase, which lengthens the telomeres in order to make sure that your gametes are always containing all of the genes that they need. So the other thing I want to go over real quick is chromosome structure. Remember that bacterial DNA is double-stranded in a circular fashion in a DNA molecule, and in a eukaryotic chromosome, it is a linear fashion. So it's a circle in bacteria, and it is linear for eukaryotes. In bacteria, DNA is supercoiled and found in a region called the nucleoid. In your eukaryotic cells, it is a complex of both DNA and protein called chromatin. And the chromosomes fit into the nucleus through an elaborate multi-level system of packing. And because of that, we're able to condense the chromosomes or spread them back out. Proteins called histones are responsible for the first level of DNA packing. And then essentially it's a kind of wrapping around of the chromosomes around these histones, and then the histones themselves wrapping around that is able to pack in the DNA very tightly. And so you can see the process here. So you've got your DNA, which is double helix. Those double helixes wrap around some histones. The histones are then wrapped around each other. And so what you end up with is a chromosome that is very nice and compactly packed rather than being very spread out.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes.
So the dense packing of the chromatin makes it possible for the molecular machinery largely to get to it. So the chromosomes are kind of dynamic structures. They're not one thing or another. They change a lot. And so when they're loosened, they allow for the replication to take place. They allow for transcription and translation to take place. When they're tightly packed, that they're not allowed to do that. And so the chemical modifications can occur that allows the chromatin to either be packed in tight or to be loosened up, and that matters in terms of regulating gene expression. And we'll talk more about that in later units or later videos. All right, that's all I've got for you today. Thanks so much for watching, and I'll see you next time.